Hello to every researcher. As we have here today to talk about the DeepFrag, which is actually a web server-based platform and basically used for the optimization of the legal. So, what does actually uh, what does actually the server is and how does it works? We will see in this presentation. There's a deep learning based tool which is designed to assess in lead optimization and crucial state with early stage drug discovery. Because in every drug discovery, we have to have a lead where a number of functional groups. So we want to optimize this functional group, we can utilize this web server. It uses CNN, which is convolutional neural networking, and it analyzes the structure of the protein receptor and the bound ligand because the ligand has to bind with the receptor with the proper affinity, which is actually the overall base utilizing this particular web server. It is recommended the chemical fragments that can be linked to the ligand to improve its binding affinity. And once the binding affinity can be improved, well, definitely the lead can definitely be optimized in the lines. Now, it's developed by the student now. It helps the researcher to identify the optimal modification of the protein, and it is acting as a virtual medicinal chemist for the hypothesis journey. As we know that in every research, we have to generate the hypothesis in order to execute the work. Now, this is all about the structure of and the visualization of the web server where you can see the frame is written, see the proper place for the uploading of the receptor and ligand, whatever you are working on and what do you want to explore. This is like the input receptor ligand file. If you want to browse, then you can simply input your protein file. It could be PDB format or it can be picture format. In the case of your ligand file, it can be PDB, it can be SDF, it can be MOL2. So these multi-format acceptability is there with this web server. You can further see the other functions of this particular server. It based upon the fragment-based optimization to suggest a specific fragment to add as a parent ligand to enhance its fit within the receptor block. And you all know that the receptor pocket should definitely optimally be accommodate ligand for a specific affinity study. The 3D convolution neural network is AI-based analysis where 3D voxel representation of the receptor and ligand complex will be visualized through the web server. It has got high accuracy, and the last thing is open sources. So you don't need to pay anything. It is open for all the researchers who are working with the computational chemistry. This is the visualization you will observe in your research. You can see the protein share along with the 3D structure of the protein. And these are the different fragments which are attached with it as in the molecular viewer. You can also observe this kind of web page where once you have uploaded your protein and the subject, you have to directly click on start DeepFrag where it will work. And after once it will completely over with whatever a protein and ligand uploading and their completely fragmentation has been done. We'll observe the result in this way. So you can see the spines, the structures and their specific score. The ranking talks about the highest affinity of particular structure of the lead with protein and how the ranking will definitely be available when the study is done. So let's see what is it and how you can utilize it. So for this particular thing, as mm, I'm directly showing you the web page. Let me leave the file for you so that you can have time to come by yourself. So you simply have to Google it, DeepFrag. You will find this particular website, studentlab.fin.edu, and this will be the model. Once you will do this thing, you will find this web page where there is the research publication, the people, the software, the teaching tools, which are, and you directly click on the web application. So let me see whether it is capturing it fully or not. So yeah, show a little bit. Wait for a second. Okay, so this is a particular web page which is of DeepFrag. How does it look like? This is the way it looks. So only have to enter durantlab.fit.edu. You will be able to search it through search engine. And once you will be able to get it, you only have to click on web application. Once the web application page will appear, you will see the receptor and the ligand file where you can treat the receptor as a PDB as QR format. In the case of ligand, you have the freedom of using PDB, MOL, SDF, all these formats can work here. Only have to browse it, give the path of your receptor and ligand file. You can even, for the first time, you can even use the example. So for today's presentation, I'm going to use example 5. You can see this is the example 5 where the ligand is attached with a specific protein. And the ligand which I'm going to use is carboxylate pointing, and the protein which I'm using is peptidyl prolysis transisomerase. Its PDB ID is 2xP9. Now, once everything is done, you can select the item, the going point. You can select the rotation. It is the default setting, so I'm not changing it. If according to your research paper you want to change it, you may even go for it. Now you only have to click over here, then you start the fragmentation. Once you will click over here, it will take some time because it's a web server, and you know once you're going for any or giving any job, there are queues for the jobs. So it will done in fraction of second. So see, while I'm simply talking about it, it is done. Now this is a structure which you will observe through the visualization, molecular visualization pattern. And you may even observe the different scores which are there. So these are the ranking, these are the smiles, these are the structures, and these are the respective scores. So as we have discussed earlier, higher is the ranking, better is the score. So that's how you can even say, like, suppose if you want that this function group in your molecule is good, you can choose it and you can go for the other side of the molecule where you can replace this specific functional moiety with the other, and then you can again go for structure based optimization of your ligand. That's how this particular thing will work. So I hope like you will utilize this server for your research work. Thank you everyone, and continue your research.